In this video, we will continue our discussion about the C1GalT1 knocked out in the uh, pancreatic uh, pancreatic cancer affected mice. So in this case, we have let us just briefly provide a overview what we are comparing. We are comparing two mice models. One mice model that has these general characteristics which are found in the pancreatic tumors or pancreatic cancer. That is, it has a KRAS mutation, K and TP53 mutations are also there. And in the other case, we have all these mutations. Along with that, we also knocked out the C1GalT1, and this kind of mice models we call them as KPCC mice models. So in this figure, we will discuss the results about the change in the glycosylation because of this C1GalT1 knocked out. So this panel 1 is about the panel 1 reflects or presents the results about Alcine blue. This Alcine blue is usually used to detect the presence of silated glycans. So in this figure on the right hand side, we can see that this C1GalT1 helps in the addition of galactose to this GalNAc monosaccharide, and after that the final product that is formed is silyl core 1, and this is also known as T carbohydrate antigen, and it has this sialic acid structure. So first we need to check whether this C1GalT1 results in the loss of silated structures or not, and this thing that we have validated using this Alcine blue staining. It usually detects or provide an idea about the presence of silated glycans on the samples, and we can see that in the case of KPCC, there is a reduced indication of this Alcine blue compared to the KPC or the control mice model. And these things are further when they are quantified, these results are quite evident that in the case of KPC, the presence of the silated glycans are more compared to the case of KPCC. And thereafter, so from this we can see that this kind of structures might be less, and in the case of this KPCC. So in order to validate these structures, that is T carbohydrate structures, we further did what we call this histology analysis of this KPC and KPCC mice model in the and using this PNA lectin, which is used which has a specificity towards this silyl core 1 glycan, or in other terms this T carbohydrate antigens. And we can see that in the case of KPC there are more presence of this silyl core T1, and whereas in KPCC there is less presence of the silyl T1. And when we quantified these images, we can see the difference is quite evident, and it is significant. Difference is statistically significant. And thereafter we also did the fluorescence assay. Apart from this histology analysis, we also did the fluorescence assay, and there also the results are in line with these histology results. That is, KPC has more number of these T carbohydrate structures compared to this KPCC. And so right now they have compared these T carbohydrate structures. However, they want to know whether there is a upregulation of this silyl TN antigen because of the absence of the C1GalT1, whether this TN or this monosaccharide is diverted towards the silyl TN structure glycan formations or not. And for this they did the histology analysis using VVA lectin. VVA lectin is specific towards the silyl TN antigen, and here they can see that the presence of this VVA the VVA signals are quite high in the case of KPCC compared to the KPC mice models. And the quantitative results shows that the difference is significant or statistically significant. And they also did the fluorescence assay of this They also did the fluorescence analysis of this VVA lectin, or in other terms, to check whether these TN antigens are present, or the difference in the TN antigen in both the models. And they can see that in the case of this KPCC or C1GalT1 knockout mice model, there is a high fluorescence compared to the KPC mice model, and this further indicates that in the case of KPCC, there is an increase in the TN carbohydrate antigens, and these TN carbohydrates antigens are usually the biomarkers of the tumors, and they are found in more more abundant amount on the tumor cells or on the membrane of the tumors. So this studies indicates that C1GalT1 could be the one of the main cause for the aggressiveness of the tumors within the patients. So I hope you have understood how this C1GalT1 is cause the alteration within the glycosylation. And if you need to understand or know more about the C1GalT1 impact on the pancreatic tumors, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.